Hi everyone, welcome to my video about protein synthesis. Okay, we're going to be talking about in this video how we actually take that DNA to make proteins, such as hormones or enzymes, okay, depending on the sequence of amino acids that we're creating. So in this process, you guys have this diagram in your notes that you're going to be using to fill in. The first thing that we're going to be talking about is transcription, the first step of protein synthesis, where we're taking the DNA in the nucleus and we're photocopying it in the form of mRNA, messenger RNA, and then the messenger RNA is going to leave the nucleus and it's going to meet up with the ribosome inside the cytoplasm, and this is where we're actually going to take this mRNA, the photocopy of DNA, and make proteins out of it. So our first step is transcription, and then the second step is translation. So the first thing that has to happen in transcription, okay, when we're making a photocopy of DNA, is we have an enzyme called RNA polymerase. It's going to unzip and unwind DNA, just like we did in DNA replication, so that we can get in there and add the complementary strand in the form of an mRNA, just replacing the T's with U's. So in order for us to be able to make sure that we're putting in the proper letters in the right place, we want to put it on the inside of the DNA strand instead of copying it on the outside. So we're going to open up the DNA strand in order to do that. So RNA polymerase is going to go along and open up our DNA. Okay. Again, this is happening in the nucleus. You guys have this diagram again, but don't write anything yet. I'll tell you guys when to start writing stuff down. So after that goes ahead and completely unwinds and unzips our DNA, it's going to look like this. Okay, I'm going to zoom in on the nucleus just so we can see it a little bit better, because there would be letters included in that DNA strand. And the second thing that needs to happen is we need to take that same enzyme, RNA polymerase, and it's going to go along and add nucleotides of RNA. So nucleotides as in it has a phosphate, a sugar, and a nitrogen base. The only difference is this time the sugar is going to be a ribose sugar instead of deoxyribose. And the nitrogen bases include, instead of thymine or T's, we have uracil or U's. So RNA polymerase is going to go along and copy our DNA, making a complementary strand of mRNA, seeing that the letters match up. So T's match with A's, A's are going to match with U's instead of T's, C's are matching with G's, G's are matching with C's, etc., so that we have a photocopy of a piece of DNA. Okay, so that is all that we are doing in transcription. We're just rewriting our DNA in the form of mRNA so that we have a photocopy of it, so it's free to move throughout our cell. So it's going to look like this, and you guys can draw this now in your diagram in your notes. So having the DNA separated, okay, and the mRNA being built on the inside. So now we're going to move into our second process that's in protein synthesis called translation. So this time we're taking our DNA and we're going to change it into the protein language. Okay, so making an actual enzyme or a hormone or a protein that's on the cell membrane that allows things inside the cell or out of the cell. So the first thing that we have to do is we have to put this mRNA strand, remove it from the nucleus, and allow it into the cytoplasm and enter into the ribosome. So it's going to slowly move into the ribosome, which looks like a cheeseburger. So that is the first step to translation. Okay, after that happens, then we're going to look at the mRNA codons, and we have tRNA, transfer RNA, that have an amino acid attached to the top of it. Okay, and they're going to have an anticodon, so letters that pair up with the codon of mRNA, so they know that they're supposed to attach in a certain place, and based on the mRNA codons. So we're going to have a tRNA molecule looks like this, okay, carrying an amino acid on the top. This amino acid happens to be the start amino acid, which we looked up in our table using AUG. Okay, AUG either is an initiator codon, or it also can code for methionine if it's found in the middle of a protein. So because it's at the very beginning of this protein, it's going to be a start. So we're going to have this tRNA molecule move into the ribosome unit, okay, and that is going to be the beginning of our protein. The anticodon is just a set of letters on the tRNA. It just pairs up complementary to the codon, but we don't use it to find the amino acid. Okay, so make sure you're not looking up anticodons in your messenger RNA codon table that's on page 7 of your notes. So the next thing that's going to happen is we're going to have the next anticodon with tRNA match up with our next mRNA codon. So this time we're looking for CGU, which in your guys' table tells you that it's arginine. So this tRNA carries arginine, and it's going to slip into the ribosome. And then what's going to happen is the amino acids that are on the top of the tRNA molecules are going to start to form bonds between them to eventually build a protein. So we just have a bond formed between the start and the arginine. And then we're basically just going to keep doing that until we reach a stop codon. So in this one, we have a really short little piece of DNA, so we'll reach the stop codon fairly soon. Okay, so the next piece of mRNA says AAC. Our codon is AAC, which matches up with asparagine. So this asparagine on the tRNA is going to slip into our ribosome. Okay, and the same thing's going to happen. We're going to start building our chain by building bonds between the arginine and asparagine. And then we're going to shift the ribosome over. We are going to grab the next tRNA molecule, which happens to be a stop, and we're going to add the stop to our little piece of our little protein. So once that is done, okay, normally your protein would be significantly longer than this, but just a short example so you guys can kind of understand this process. Then we are going to send that protein to be packaged in the Golgi, which is this organelle. Okay, when it gets packaged, it builds bonds between the molecule to basically make it into a little folded up circle of protein so that it's easier to move around your body, so that it's not a straight line and getting tangled around stuff. So that is basically all that there is to protein synthesis. So make sure you guys understand this diagram, understand the processes. We will review a little bit more in class tomorrow, but if you have any questions, please ask me. If not, I will see you all tomorrow. Have a great evening. Good night.